Please introduce ma'am, Rinku ma'am.
Okay. So a very good afternoon to all of you. Today we are on the day 5th in the afternoon session of the 21 days online national training program on advances in veterinary research for sustainable development of livestock sector. So we are very happy to have with us Dr. Heena Sharma. She is a scientist working in the dairy technology division of NDRI Karnal, and I would like to briefly introduce her. She has been serving the Indian Council of Agricultural Research as a scientist, and Dr. Hina, she holds expertise in the area of fermented and functional dairy products from small ruminants, milk, health beneficial effects of herbal dairy products, metabolites generated during processing and fermentation. Her research interest area is related to the development of functional milk products from non-bovine milk and milk products including goat and camel milk. She has been associated with several projects as a PI as well as co-PI, and several technologies for her research group have been approved for commercialization related to goat milk cheese rolls, goat milk probiotic yogurt, and functional goat milk beverage. She has the accuracy to handle sophisticated and advanced high throughput instruments. Dr. Sharma has around 25 research papers, 6 review articles, and 15 other articles in her account. She has received several awards, poster, as well as oral presentations in national and international conferences. Dr. Sharma has attended several training programs in the area, including probiotics, value addition to dairy products, basic application of metabolomics, and also delivered lectures in training related to milk processing and value addition. She has obtained international fellowship for 5 months international training at Oklahoma State University, USA, wherein she worked on metabolomics. Recently, Dr. Sharma has received NADSI Associate as an excellence of a contribution in research and dairy science. So I would like to now invite Dr. Heena Sharma for her lecture today, which is entitled as Bioactive Compounds in Functional Milk Products. Welcome, Dr. Heena, please.
Thank you, Dr. Rinku. Thank you so much. I also thank the organizers for inviting me to express my views and interact with you all. So I shall present now. The topic, as told, it is bioactive compounds from functional milk products. So we'll start gradually from what are functional milk products, why are they required, what do they do in our body, to the identification of the bioactive compounds which are present in the fermented food products. So nutrition and diet, or you can say nutrition and health, they are very, it's a complex mechanism which is closely related, and it reduces, if we are not taking enough nutrition in our body, it reduces both cell mediated as well as humoral immunity. So all these nutritional inadequacy in our diet, it leads to gut dysbiosis, means there is imbalance of the microorganisms in the gut system. There will be sarcopenia, loss of muscle waste, muscle growth, skin atrophy, physiological stress will be there, reduced mucosal functions. So all these are well known and very basic mechanisms which undergo when we are not taking any proper diet. So coming on to what is immune system, whenever we think about functional foods mainly related to probiotics or herbs incorporated in the product, or any other bio and vitamin enriched products. So whenever we think about that, we always think about the immune system, that what it is going to do to our immune system, because immune system is the basis for our healthy body. Defense against microorganisms, defense against pathogens, invading microorganisms, all these lies on the shoulders of immune system. So immune system is basically the body's defense against disease causing organisms, malfunctioning cells and foreign particles. So what types of immunity we have in our body? It's very general. We have active immunity, we have passive immunity. What is active immunity? Actually, active immunity is the first line of defense, that is the innate immunity, and it is developed when our body is exposed to an antigen, so that an adaptive immune response is generated. So usually if we talk about active immunity, it is long lasting. It generally develops and it lasts till weeks, days to weeks. And if we can tell that it is of two types, natural and acquired immunity. Natural, that which is existing in the infants, in the newborn babies also, who do not have any prior sensitization to antigens or foreign particles. Whereas acquired immunity, it develops when a person's immune system is counteracted with the foreign substances. So we have two types of active immunity here. Whereas passive immunity is developed when a person is given antibodies to a particular against a particular pathogen, like we are getting vaccinated for COVID-19 these days. So it's kind of a passive immunity which is developed with the time, or when we are getting antibodies against that particular pathogen. So that is the passive immunity. Now all these immunity, active immunity, passive immunity, we are taking vaccines, we are exposed to foreign antigens. What are they doing? They are actually, in nutshell, they are producing antibodies, they are activating our cells in the immune system, that is T cells, B cells, interferons are increased, macrophages are activated. So all these, in nutshell, is because of our response to the foreign invading pathogens. So if we talk about innate immunity and adaptive immunity, as I have already told you, different types of cells are activated. In case of innate immunity, if we see, we have macrophages, dendritic cells. These are mainly the antigen presenting cells. Then we have leukocytes, neutrophils, eosinophils, basophils, all these are activated. While adaptive immunity, we have cells, T cells, B cells, and antibodies production. Natural killer T cells, these are common in both. Either we have innate immunity or adaptive immunity response, natural killer T cells are produced. So coming back to the inadequate nutrition, definitely our hectic, stressful lifestyle, our workload in the offices, at the home, fast food, energy dense food, but there are micronutrient poor food. Because of this, we often neglect the micronutrients in our diet, like iron, sodium, or you can say calcium. So sedentary lifestyle, we are not doing any physical activity regularly because of overworkload, that is leading to obesity conditions. So all these are poor diet conditions. These are leading to suboptimal nutritional status in the society, and then we are having compromised immune functions. So compromised immune functions, definitely it will lead to the increased risk of infection. So it depends from person to person how our immune system is responding, and that depends on our diet mainly. So in this regard, functional foods, they have a major role to play. So what is actually the functional food? What this word functional means? Functional is actually when we are adding an ingredient to a food or milk product, meat product, any food, so that it becomes functional, so that it performs a specific biological role. So functional food is a food given with the additional function. That is why we call it as functional food. So natural or processed food which contains known biologically active compounds. Why the term biologically active compounds is very important here, because there are so many compounds generated in the food. When we are processing, even with the heating of the milk, all the constituents, almost 70 to 80% of the constituents are changed as compared to the raw milk or fresh milk. So these biologically active compounds are necessary to be absorbed in the body, so that we can have they can exert the specific biological function in the target organ or in the target tissue. So these are usually in the defined quantitative or qualitative amounts which are present in the food, and they provide a clinically proven and documented health benefit. So there are a lot of researches, extensive research work is going on the functional foods, just because now people are more and more they are relying on their diet for the healthiness, for the health and the well-being of the individual. So it includes processed foods or foods fortified with health promoting additives like vitamin enriched products. We also have probiotics, like we have Yakult in our in the market. That is the first commercial probiotic dairy product which is available. So all these products, these are functional foods which perform a specific function in your body when upon ingestion. So why there is an interest in functional foods? As I have already mentioned this, rapid advances in science and technology, mainly the biotechnology, nutrigenomics, Now the researchers are trying to correlate the genetics or genes expressed in the foods, or you can say during fermentation or during ripening process of cheese, whatever the genes are expressed, or upon ingestion of these products, whatever the genes are expressed, these are correlated with the nutrition of the individual. Suppose somebody is consuming this Yakult daily. So we want to know what all genes are expressed or what all genes are inhibited during its during the process of consumption, so that we can know how these genes are related with the health of the individual. Likewise, biotechnology, if I take an example from this science, bacteriocins are produced from the bacterial fermentation, during the bacterial fermentation of milk products, and these bacteriocins are now used as natural preservatives. So it's a breakthrough research in science and technology that we are getting the natural food for eating. Then there is evidence linking diet to chronic disease risk. Then we have a desire to attain wellness through the diet. We don't want medicines. We don't want those vaccine injections. Then aging population also requires a specific diet. That is why we have some particularly designed functional foods. Then healthcare costs are also they are skyrocketing. That's why we need an alternative to a health, that is through our diet. Then changes in the food laws, label and product claims. Now, what are nutrients that are present in the food which support or improve the response for our immune system? We have major components. We have proteins, we have fat, vitamins, minerals. Then we have antioxidants and bioactive compounds. Now, what are these bioactive compounds? I'll deal this within subsequent slides. Before that, let us focus on what are source of functional foods. We have n number of sources available. Then we have animal source, we have milk, meat, fish. Naturally occurring, we have fruits, vegetables, or fruit juices are there. Then plant source, we have legumes as very good source of functional foods. We have wheat, buckwheat mainly. Then we have symbiotics. What are symbiotics? This is nothing but the combination of probiotics and prebiotics. That is known as symbiotic. And why do we add prebiotic? Prebiotic mainly acts as a base for the growth of the probiotic, as a supplement for the growth of probiotic. Since whenever we want to consume a probiotic product, we ought to have minimum number of viable cells in that product upon ingestion, right? So for that we need prebiotics so that it can supplement the growth of probiotic. That is symbiotic product. Then we have industrial products, like in market we have so many, Dabur is there, Patanjali is there. Then ultimately we have nutraceuticals, that is the supplement, food supplements, vitamin B, vitamin D, all naturally extracted. These are processed in the form of supplements, so we can have the functional foods from these sources also. So one major breakthrough research in the functional milk products was the composite dairy foods. Now, why do we need composite dairy foods? These are usually the formulated foods which has significant amount of two different food groups. Suppose we are mixing pulses with meat. I'm talking about meat products here, pulses with the meat products. And then we have dairy foods with some wheat or barley or bajra. So all these supplement the micronutrient requirement usually. So these are usually unrelated food groups for the development of new product, solely driven by deriving a new flavor, and complementation for organoleptic, functional and health virtues. So why composite dairy foods? Why not only the milk? Because milk lacks certain micronutrients and fiber, as we all know. So milk alone, it cannot enhance the acceptability of developed products also. So just to enhance or improve the sensory acceptability or overcoming the challenges of developing new functional food product, we go with the admixture of milk with some composite foods like wheat flour or some barley, bajra, like that. So supplementation with micronutrient and health promoting component, it will definitely address to the issue of inadequacy in the nutrition. So, what are promising candidates for these composite dairy foods? We have cereals, grains, flour, barley, oats, and we already consume this kheer. In the Indian society, it is majorly consumed. Malted milk foods are there. So, the all available in the market, the Bournvita, Horlicks, all these are malted foods. When we consume it with the milk, that is malted milk foods. Then we have millets, coarse millets are there, which are rich in calcium, iron, phosphorus, zinc. So we get the micronutrients when we combine it with the milk or milk products. Higher proportion of non-starchy polysaccharides are there. Dietary fiber is there, which is very much required in our daily diet. Low glycemic index, it does not lead to the production of any higher amount of glucose. It also leads to the lowering of total cholesterol and low density lipoprotein cholesterol. Then apart from this, we have fruits and vegetables, usually which are lost in the post-harvest management. We use this, like peel extracts are there, or mango peel is there, carrot peels are there. All these are usually utilized for developing composite dairy foods. Then dairy products, whey, whey proteins, isolated whey proteins. Then we have whey protein concentrates. They have very high biological value and very easily digestible and very good source of proteins. Then whey powder is also utilized for the production of lactose here. They have superior functional properties like gelation, emulsification, and whipping. These are very much required for the production of products like yogurt, where we need gelation or where we need a whipping ability of the product. So all these ingredients we can use to develop the composite dairy foods. Now, what are the major challenges which we face during the when we are going to combine dairy or milk with some other food group? So numerous reactions of non-dairy components with milk molecules are usually investigated. Cereals, fruits, vegetables, they usually destabilize the milk proteins because we know fruits are having lower pH as compared to the milk. Milk is having almost neutral pH. So it disturbs the salt balance, lowers the pH, and coagulates the milk proteins. So this is a very big challenge for any researcher to develop the composite dairy food. Then presence of pigments in fruits and vegetables, thick pericarp of foods results into the poor color, so that we need to have natural colors in our food product, so as to increase the appearance of this product. Then millets contain certain anti-nutritional factors which need to be removed before adding them into the dairy product. Non-availability and short shelf life of primary processed products like grapes, flakes, semolina, and flour, these need to be addressed. And polyphenol interaction with the milk. This polyphenols usually we get in the plants. So when we are trying to add any herb into the dairy products, we usually see the polyphenol interactions with the milk, and it leads to the coagulation. Even addition up to you can say 7 to 10% or 15%, that will lead to the coagulation if you are going to produce some sterilized product, sterilized milk. So that affects the bioavailability of the proteins also and stability of the mix. Maybe suppose your fresh sterilized milk is having the shelf life of 4 months at the room temperature, but if you are adding herb and you are not stabilizing that mix, the shelf life you may get up to only 30 days. So that is the challenge, main challenge for us. Then we have addition of starchy material, which increases the viscosity of the mix and affects the thermal processing again and mechanization. So these are the challenges which are to be addressed while composing the dairy composite food. Then next is probiotics as functional food. What are these probiotics? These are live microbial feed supplements which beneficially affects the host animal by improving its intestinal microbial balance. So these probiotic products, they should have viable bacterial count. That means bacteria should be live at the time of ingestion, and that recommended value is around 10 log 7 CFU per gram of a product, so that health benefits can be accrued. Fermented foods like dairy foods, they are commonly used as probiotic carriers. And what are the characteristics of a good probiotic? It's not that any microorganism we can use as a probiotic. There are a number of microorganisms there, including pathogens. So we have to look for a good probiotic, and the qualities should be strain specific, which is capable of exerting a beneficial effect. Then it should be non-pathogenic and non-toxic. It should be present as viable cell. That is the prime most important requirement for any probiotic product. It should be capable of surviving and metabolizing in the gut environment, example, resistant to low pH and organic acid. Because we are having low pH in our gut system, we have lots of organic acid production in our gut system. So we need to have probiotic which resists all these changes. Then it should be stable and capable of remaining viable for long, because we are not, when we are producing or when we are manufacturing a product, we should have always in our mind the shelf life of the product. So because it is going for a sale, we are not consuming it on that single day of the production. So it should be stable and it should be capable of viability for longer period of time. Then in a nutshell, I'll tell you the health benefits of probiotics. It's known to all. We are listening these probiotics from very long time. So protection against infection, enhanced nutritional value, improved lactose digestion, beneficial for cardiovascular diseases as it lowers the serum cholesterol level and leads to the deconjugation of bile salts. Immunomodulation by providing a strong innate immunity, by increasing the interferons or you can say interleukin activity, better calcium absorption, prevention of osteoporosis. Then it leads to the suppression of carcinogenic substances or the organisms which leads to the production of carcinogenic substances. Then it regulates the gut motility and improves the constipation or any inflammatory bowel disease. Then improved gastrointestinal health. So this is how the probiotics usually act in the system. There are three main mechanisms if I talk about in summary. Competitive exclusion, I have covered it, yes, it's here. We have colonization resistance first of all. Colonization resistance is nothing but probiotics, they do not allow the colonization of other pathogenic organisms on the intestinal lumen, or you can say in the villi of the intestine system. Suppose here if you see this, this is pathogen, it will competitively exclude the pathogen from attaching to the intestinal mucosa. So the probiotic, it is having its own niche so that it does not allow any other pathogen to get attached here. That is the colonization resistance, or you can say competitive exclusion of the pathogens. Then second is it maintains the barrier function. It will tighten the junctions of the intestinal cells. Okay, all the villi, they will, all the cells, they will be adhered tightly to the each other so that no pathogen is able to invade the microvilli or the villi of the intestinal lumen. So it reduces the macromolecular permeability and bacterial translocation. Then we have modulation of innate or adaptive immune system. That is because of the increased immunoglobulin A, immunoglobulin G and M production. It increases the activity of anti-inflammatory cytokines like we have interleukin 10 and tumor growth factor. Then, and it will decrease the pro-inflammatory cytokines, that is the tumor necrosis factor alpha and interleukin 6. So this combination, the inhibition of pro-inflammatory cytokines and activation of anti-inflammatory cytokines, that will lead to the modulation of the immune response. Then it will also lead to the modulation of signal transduction. These are specific, you can say, pathways which these probiotics follow so as to increase the response of the immune system to the invading organisms. Then talking about the metabolic effects, we have the bacteriocin production. I have already told you these are the proteinaceous substances which are usually produced during the fermentation, and they exert specific functions against the invading pathogens. Then decreased pH, definitely it will lead to the decreased pH, and in case of decreased pH, usually the bacteria, they are not able to survive. Then we have quorum sensing. Quorum sensing is the maintenance of the population, bacterial population, in response to the changes. Suppose there is a decrease in the pH, that quorum sensing inside the bacteria, that will give the signal to the adjoining bacteria about the functionality, about how it should behave now. In case of decreased pH or in case of increased pH, we have certain conditions going on in our body. So that quorum sensing helps to transduce the signal from one bacteria to another. We can say in this way. So talking about how probiotics work, that I have already told you. Inside lamina propria, mucosal cell membrane, we have immunomodulatory actions. Then in lumen, we have antimicrobial activity, bacteriocins, defensins, increased immunoglobulin production, then competitive exclusions of bacteria. Now, these are the specific bacteria, or you can say probiotics, which are clinically proven for their health benefits. And they, like we have Lactobacillus reuteri, it will produce pathogen inhibitory substances, then inhibit the pathogen attachment, that is competitive exclusion. These are the bacteria. So I have just compiled the list so that if somebody wants to develop a specific product, suppose you want an immunomodulatory product, you can go with the stimulation of immunoglobulin A. You can go with the Lactobacillus rhamnosus GG. So all these strain-specific products also, or you can say the bacteria which you use in your product for fermentation, also determines the technological and functional capability of the product. So probiotic matrix. Now we can add probiotic in dairy as well as in non-dairy, both matrix. But in dairy it is usually preferred because they act as idle vehicle, you can say, for the delivery of the probiotics. And since milk is liquid in nature, it is having almost neutral pH. It provides the sufficient nutrients also for the growth of the probiotics, and it is easy to maintain the viable number of probiotics in the dairy matrix. Now, coming on to the bioactive compounds, this I have covered till now. What are the functions of dysfunctional foods? How does it act in our body? How are they beneficial? So talking about the bioactive compounds in milk mainly, we have these major compounds. We have water, protein, fat, lactose, minerals, then immunoglobulins, peptides, nucleotides, oligosaccharides, and metabolites. Now, what are these metabolites actually? This is, I would say this is not a new topic, but yes, it is recently very extensively reviewed and researched upon. Maybe because these now low molecular weight substances, which are usually less than 1500 Dalton, these have proven their significance in maintaining the technological acceptability of the product. So we have certain role of these low molecular weight substances in our functional products, functional fermented dairy products, so that the viability of the product or the health benefits of the product are usually proven on this basis. So different origin and sources of metabolites, they contribute to the variability of the milk metabolite profiles. So if you're talking about goat milk, if you're talking about cow milk, buffalo milk, or sheep milk, camel milk, yak milk, any species, there will be difference in the milk metabolites. Definitely there will be difference, because these metabolites are generally naturally existing in the body, and they are usually produced, generated upon the metabolic activity of the enzymes. So whatever the diet is animal is taking, depending on that, enzymes act or bacteria act so that the different metabolites are generated. So talking about the milk metabolome, what is metabolome actually? So I told you about the inherent factors and external factors. Like inherent factors, species wise, if you go, there will be difference in the metabolites. Breed wise, if you go within the species, if you go with the breed wise, Sahiwal breed, Tharparkar breed, or Red Sindhi breed, all these breeds will have different metabolite production because of their own metabolic activity. Lactation stage, parity, how many parturitions the cow have undergone. So all these factors combined with external factors, their feed, their season, their health, processing, storage of the milk or milk product. So all these relates to the milk metabolome, and this milk metabolome, it determines the technological characteristics as well as the physicochemical characteristics of the milk and the resultant milk product. So these are the different terminologies which are usually used when we talk about the milk metabolome or any food metabolome, you can say. Food component analysis, it's a chemical component analysis. Biomarker is a specific metabolite, or you can say specific low molecular weight organic substance that is usually identified in a particular substance. These are specific to a particular food group or any milk product, or any, if you talk about yogurt, there will be specific yogurt biomarkers. If you talk about the cheese, then there will be specific biomarkers. So then we have metabolomics, determination and the quantitative analysis of these metabolites. Metabolome, it's a collection of all the metabolites which are present in the cell, organ, tissue, organelle, like that. Then what are metabolites? These are usually endogenous and exogenous chemical substances such as peptides, organic acids, methyl ketones. But mind you, the only thing, you will say that peptides are known from like past 50 years. Organic acids are known from past 50 years. What is new about metabolites? These are the substances which are having molecular weight of less than 1500 Dalton. You cannot call all organic acids as metabolites until unless they are having weight less than 1500 Dalton weight. Now this metabolomics, it is usually used in case of the estimation of food quality, safety, processing conditions, microbiology. We call it as metagenomics, then sometimes metabolomics, then we have food nutrition and functional foods. So food component analysis, how it varied when this metabolomics was introduced. Because the traditional, it usually estimated the protein, fat, ash, water, minerals, carbohydrates, calorie content. But with this metabolomics, we can identify each and every single amino acid. We can identify each and every this mineral, zinc, iron, potassium. We can know the derivatives of fatty acids also. We can know the derivatives of proteins also, like dipeptides, tripeptides, because they all influence the texture of the product. So talking about the milk metabolomics, this is just a literature survey. We have four techniques by which we can estimate these metabolites in the product. Whenever I'm using the word metabolites, it's usually the bioactive compounds. Okay, just don't get confused that I'm talking about only metabolites, not bioactive compounds. It is one and the same thing almost, as per the functional food context is concerned. So NMR, nuclear magnetic resonance, from that we could identify almost 39 metabolites. From LCMS, 116 metabolites. HPLC, 12 metabolites. GCMS, we could go with the 30. Text mining is like the literature, or you can say the meta-analysis, it's 255. So we have these different techniques nowadays, GCMS, NMR, and LCMS. These GCMS and LCMS, these are mass spectrometry based, whereas NMR, it's a different technique, nuclear magnetic resonance. So these three techniques are mainly used worldwide nowadays to identify the major bioactive compounds or metabolites present in the fermented dairy products. So we have two approaches here to identify or quantitate the metabolites, targeted as well as untargeted. Usually when a researcher is starting the research, or you can say it's new to the field of metabolomics, like I have just started it, so I'm going with the untargeted metabolomics. Untargeted is hypothesis driven, that we know that in yogurt, definitely there is fermentation going on, there will be peptide production, definitely there will be some generation of low molecular weight substances. So we can go with the untargeted metabolomics because we know that we are not going to fail. Okay, so it's just to be on safer side, we go with the untargeted metabolomics, where identification is already known, correlated to the reference standards, and absolute quantification is done. Whereas in targeted metabolomics, we generate the hypothesis. Okay, we do not know that in this product how much of lactic acid is there. We do not know. So it's kind of a qualitative identification and relative quantification. We usually compare it. Suppose targeted metabolomics is usually used for the existing product, and when we are going to develop a new product over the existing formulation, at that time we use targeted metabolomics. So it's a one step ahead of the untargeted metabolomics. So talking about the different techniques of this identification of metabolites. So working principle for GCMS. GCMS is gas chromatography mass spectrometry. We introduce a sample, there will be ion source which will produce the ion, then there will be mass analyzer which will analyze the metabolites on the basis of their weight. Because I told you that metabolites, we will segregate all those metabolites or we'll sort all those metabolites which are having less than 1500 Dalton weight. So this mass analyzer is having important role in that. So what it will do, it will segregate all these substances on the basis of their size, and only those substances which or only those compounds which are having less than 1500 Dalton weight will go to the ion detector. An ion detector, it will give you the peaks as per the ion presence of ions in that, as per the quantification of ions, and the output or data will be generated in the output system, that the computer which will be connected to the GCMS. So this is the picture or image for gas chromatography. Here we have the ion source. We usually put, this is our liquid, you can say, sorry, sample collector. We'll put, it will come to, we will give the command and it will come to this side, take the sample, inject it inside. Here we have the syringe. Okay, there is a syringe. It just takes 5 microliter of the sample, not more than that. So we do not require a mass of sample like 1 liter, 2 liter, or even 5 ml, 6 ml. It will just take 5 microliter of the sample. It will inject inside. There will be a column. Here you can say injector. There will be a column. It will run here, and the heavy substances that they will be lagging behind, whereas the low molecular weight substances, they will go to the ion source first. And here mass filter is there, or mass analyzer you can say. These deflections will occur. All these ions will deflected towards each other. So the low molecular weight substances, they will hit the target first, whereas the heavy, or you can say the high molecular weight substances, they will hit the target late. Okay, so we usually keep the retention time. Retention time is the time which is taken by a metabolite to hit the target. Okay, so in case of milk and milk products, usually the time varies between 60 to 90 minutes, if you can say. So that means one sample will take almost 60 to 90 minutes to run. Okay. However, in case of meat products, what I have observed, based on my experience, it will take just 30 minutes. We keep the retention time of 30 minutes. So principal partition, mobile phase is gas or liquid, stationary phase is usually solid or heavy liquid coated onto a solid or support system. So this is the protocol. How do we proceed with this metabolite identification when we are having any fermented dairy product or any other product, you can say. So first of all, the primary requirement is it should be defatted. There should be no fat. Otherwise this fat will be will interfere with the separation of these bioactive compounds or metabolites. Okay, so we have to prepare the sample very cautiously. It should be defatted. Then you can go with the grinding, homogenization, filtration, dilution, whatever mechanism you want to apply or technique you want to apply, you can go with that. Then extraction with a suitable solvent. Suppose you are having a fat-rich product. Okay, how to go with that? Because you cannot remove the fat. Suppose you have a ghee. In that, how you will go with the GCMS? So usually we prefer GC with that. We hardly we go with the MS. But in case we want to go with the MS for that, we extract it with the suitable solvent. Suitable solvents for ghee, I would say it will be methanol or chloroform, so that it can give you a polar phase and a non-polar phase. So you can go for GCMS, you can go with the polar phase. Okay. So again, if you want to go with the targeted metabolomics, whether untargeted metabolomics, that depends on you. Then you have 3 techniques for this. We also have FTIR and IR. This is relatively simple as compared to these 3. Then separation and detection of the metabolites is done. That computer will give you the file in the form of peaks. Then deconvolution of MS spectra is there. I will tell you how it is done. Peak identification, comparison with the reference library, data processing and interpretation. So process for metabolomics, sample preparation, no fat, it's a big no. Then reduced sample size is there. We usually take almost 100 microliter or 200 microliter of the sample. Then concentration of the sample is also very important. It should be mixed thoroughly. A representative sample should be taken. Then use of liquid nitrogen. Liquid nitrogen is usually used when we have already extracted the sample, but we are not going to allow the extraction solvent inside the system, okay? Because we want the metabolites only from our product. So liquid nitrogen is used to dry out that extraction solvent, okay? Or we can go with the freeze drying also. But usually, just to hasten the process, we go with the liquid nitrogen. So this is the glimpse of the procedure. This is the utilization of the liquid nitrogen. Then we have this. Then extraction and derivatization. Extraction, I've already told you, we use with the suitable solvent. And then with the internal standards, like we can use succinic acid is used usually. So we also derivatize the sample with MOX reagent. It is prepared in the hydrochloride and BSTFA, MSTFA. You will get ample of literature in the internet for this. Then in untargeted metabolomics, solvent is usually optional because solvent usually interferes with the metabolite process. So we go with, when we are sure of this metabolite present in that, then we go with the solvent. Then extraction of non-volatile metabolites, it requires derivatization to increase the thermal stability and volatility. Silylation is nothing but when the derivatized compounds are usually seen in our GCMS results. That is silylation. So we should avoid that during our research. Then separation and detection of metabolites, we usually do with one of these equipments. Detection techniques are mass spectrometry, NMR, NIR, FTIR. Extraction of non-volatile metabolites, it requires derivatization again to increase thermal stability and volatility. Then data processing and analysis, generation of huge data. Huge data is generated because you will get around thousands of metabolites in one compound. Just in 100 microliter of your sample, then 5 microliter of that sample which went into the GCMS, you will get around thousands of metabolites, okay? So you will deconvolute MS spectra. Deconvolution of MS spectra is, there will be peaks when you get the results. So you want to deconvolute it. You want to interpret those peaks, that particular peak is related to which metabolite. Elimination of the outliers, normalization of the peaks and compound identification and peak identification. Spectra, then data set, multivariate analysis or univariate analysis is used. Now this is the one of the softwares which Agilent GCMS is using to deconvolute. It's AMDIS, usually we call it as AMDIS, Automated Mass Spectral Deconvolution Identification System. So the files are generated in the form of .d format. We will select this file and then we have this. This is the first page of the software. When we are selecting the file, this is the peak. Suppose you are having these peaks in your results. It's a sample of yogurt. What I remember, it's a sample of yogurt. So here the deconvolution has taken place, okay? It has given that the retention time is here 57 seconds, 71 seconds, 85 seconds, like that you will have n number of metabolites. Then you will, if you're interested in a particular peak, you can see what are metabolites present in this particular peak. So one particular peak is composed of n number of peaks. That is why I am telling you just 100 microliter or 5 microliter of sample will have thousands of metabolites. So now it depends upon your interpretation of results, how you want to proceed. Suppose you have yogurt with you and you have kept it for storage, okay, or you have cheese and you have kept it for ripening. So you want to see the changes during the first month of ripening, second month of ripening, third month of ripening, and you are particularly interested in proteins. So you will go, suppose this 43.73 retention time, this is related to some dipeptide, leucine, isoleucine, okay? And in this you want to see what are other metabolites are present. So you will get n number of amino acids here. So this is how these results are interpreted. So this is another method. Here you are matching this NIST library, in which you will get to know the exact name of the compound. So here you match comparison of the two samples, and once you have already with you the abundance of the samples or concentration of the concentration of the metabolites, then you go with this Metaboanalyst software. This is a free software available on the internet. You can play around whatever you want. You want to see statistical analysis, multivariate analysis, univariate analysis, then your enrichment analysis, pathway, joint pathway, whatever you want to do, you can do with this. Targeted or untargeted metabolomics, you want to go with this. If you want to see untargeted metabolomics, you want to go with this. So it's very friendly, user-friendly software. So here you have certain formats in which you have prepared your Excel sheet or data file. Then the results are interpreted like this. This is the enrichment analysis. It will tell you, upon clicking this circle, you will get to know that this particular metabolite is involved in which cycle. Suppose pyruvic acid is there. It will tell you in which step of glycolysis pyruvic acid is more upregulated or downregulated. So this is a network how all these metabolites are related. Suppose you have here, I think these are hundreds, thousands of metabolites. So how these are related, and if you are interested in one particular metabolite or one particular pathway, you can just extract these files. All these are upregulated, downregulated. So there are a number of work, researches are going on in this field, metabolite differences in relation to dairy species. If you see, they have compared the metabolomics comparison between sheep and goat milk, and they have revealed why goat milk is important for infants or why goat milk is easily digestible. So as far as the traditional analysis is concerned, we knew that goat milk is highly digestible because it is having smaller fat globule size, right? But this metabolomics has told you a new thing, that which particular fatty acid is more present, or which particular low molecular weight substance is present, which is accruing this property to the fat for its smaller size. So here if you see goat milk, it is having increased mannose-6-phosphate and fucose, increased valine and leucine. So these mannose-6-phosphate and fucose, if you know that these are very important for the brain development. That is why goat milk is usually recommended for the infants. Sheep milk, it's having higher amount of alpha-ketoglutarate and myo-inositol. This myo-inositol is the, you can say, good for the healthy gut system. So if you are having a disturbed gut system, you can go with the sheep milk. At least you have a scientific basis why. So this is what metabolomics or metabolite profiling does. So in this case, metabolomic biomarkers, they identify differences in the milk produced by Holstein cows and other minor dairy animals. So I told you, within the species also, you will have the breed differences, breed specific differences. Here you can see York milk, higher concentration of this histidine but lower amount of succinate pyruvate. So we know that glycolysis, the metabolites involved in the glycolysis are lesser in case of York milk. So this is how the results are interpreted, and they form the basis for the development of the functional milk products. Here if you see metabolite differences with respect to breeds, again, brown Swiss cow, higher levels of alpha-aminobutyric acid. So alpha-aminobutyric acid, it's higher but lower levels of ornithine. These differences may be associated with both the breed and feed management. So whenever we are doing the research, we have to keep the variables to the minimum. One or two variables will help us to interpret the results efficiently. So with respect to breeds, again, here they have used the NMR analysis. Jersey milk, silylated and complex neutral fucosylated oligosaccharides are there. Here we have simpler oligosaccharides. So for the digestibility, definitely HF milk will be preferred because it's having simpler neutral oligosaccharides. So FTIR spectroscopy is also used for this. It will give you the major compounds which are having the different peaks, absorption spectra. Here if you see this Jamnapari, Shannon and Toggenburg, the goat breeds, they were distinguished on the basis of the FTIR analysis. Now how these this metabolomics is relevant in controlling the technological properties? It's the cow milk, it affects its coagulation properties. It's well proven. A higher amount of choline and lower amount of carnitine, lactose, and citrate are required for a good coagulation properties. Now, for cheese preparation, we definitely need the coagulation. So, what are the basis or what is the mechanism behind this? Post-translational modification of the kappa casein. Kappa casein is present in the milk, which is modified post-translational, and this leads to the higher choline and lower carnitine, lactose, and citrate content. More than a single compound is also involved in the coagulation capacity of the milk. This was really revealed by this metabolite profiling during the coagulation process. Yogurt fermentation characteristics, these were also investigated using NMR metabolomics, and pasteurized milk samples exhibited stable metabolome during the initial storage. However, later pantothenic acid and butyryl carnitine decreased. So, how this is relevant? Pasteurized milk sample, it is having a shelf life of 5 days. So if you are consuming the pasteurized milk on first day or second day, you will have the stable metabolome. There will be not much change in the vitamin or mineral content. But if you are consuming the pasteurized milk of 5th or 6th day, definitely it will have decreased vitamin B, pantothenic acid, and butyryl carnitine. So these are all having specific biological roles in our body. So, in goat milk products also, fermented goat milk products also, there have been few researches. In this, GCMS technique was used to investigate the changes in the goat milk yogurt during storage. So, the authors have found that these are the exclusive metabolites which are present only during the day 14 and day 28, and listophosphocholine between day 28 and day 14, and all these are, you can say, common. So it depends on the storage duration of the yogurt, that which compound will be higher, so that when you consume, how it is going to function or how it is going to act in your body. And in this, GCMS was again used to compare goat milk yogurt and cow milk yogurt, and they have found almost 163 different metabolites as compared to goat and cow. In this, they have compared the sheep and goat yogurt, and again they have found so many metabolites which are only available in case of yogurt. So, at the end, I would say that this is the very good opportunity for researchers and scientists to further explore, advance, and contribute to the existing knowledge by, I would say, targeted metabolomics. Then, food scientists, they need to more precisely follow the consequences of different preparation and preservation processes, because ultimately, if we are going to freeze the product or if we are going to store the product, the stability of the product is going to decrease. So we should know up to what storage period amino acids will be highest, fatty acids will be highest, or you can say mineral content will be highest. So all these researches are very important in today's scenario. So understanding the complexity and diversity of various foods and to explore their functionality. Thank you.
Thank you very much, Dr. Heena, for a very interesting lecture. Before thanking you formally, I would like the participants to ask questions. At present, I am not seeing any question in the chat box. So, if anybody wants to ask the question directly to Dr. Sharma, they can ask. Please. We would welcome any question from the participants. Any one of you, please. Dr. Anil Deka is asking, what is metabolomics?
I have already mentioned metabolomics is the science now, a newly emerging science, I would say, which will help us to identify and quantify these low molecular weight substances, which are usually helpful in controlling the technological functional properties of the food products, I would say. Right.
Okay. Then next question is from Dr. Suresh. He's asking, humans or animal guts, they have beneficial microorganism. Then what is the use of probiotic intake?
Probiotic intake, okay. Probiotics, they help to maintain that healthy system. If you are consuming junk food one day or second day, maybe you will have some dysbiosis, imbalance of that, I would say, microbial balance. So just to maintain that, to restore those healthy beneficial organisms, you need to take this probiotics. Definitely nobody takes probiotics daily. It's usually taken supplement when you are having some diarrheal infection, or you had some diarrhea or fever. At that time we usually take these probiotics.
Okay. Then Dr. Pavan is asking, is there any commercial prebiotic for dogs and bovines?
Dogs and bovines commercial prebiotics, sorry, I do not know this.
Okay. Then next question is from Dr. Lalza. She is asking, what is the future prospect and is there any side effect?
Side effect for, sorry?
For probiotics, I suppose. Yeah.
Oh, no, there is no side effect as such. But again, I would say nobody takes these daily. And future prospect, I would say we have explored lactobacillus species very much for probiotics. So now the scientists are looking for some other species or some other strain so that it can serve as a good probiotic.
Okay. Then next question is from Dr. Manoj. He's asking, what is the effect of adding NaCl or common salt in milk on our health?
On our health, uh, NaCl, if you're adding NaCl in milk and you're boiling it, I would say it will rather coagulate. If you are boiling the milk, there is no effect on health. Scientifically, it's not proven, but definitely it will lead to some disbalance of mineral.
Okay. Then Dr. Papuri is asking, what is a metabolomic profiling process, how it differs from various bioactive compounds?
Metabolomic profiling process.
How it differs for various bioactive compounds?
Okay, nice question.
Yeah.
Here, I'd say if you want this, like I told you, if you want to go with the fat-rich dairy product, definitely you will have to use the different extraction solvent. If you want to go with the higher peptide, if you want to go with the estimation of peptide content or tripeptides, dry peptides, you will have to vary with your extraction solvent. Then comes the derivatizing agent. Derivatizing agents are also two types, BSTFA and MSTFA. So MSTFA it is usually used in case of milk products because of its inherent composition, and BSTFA it is mainly used in case of meat product. So with every bioactive compound and with every raw material you are using for estimation, definitely it will vary.
Okay, Dr. Pavan is asking examples of prebiotics.
We have a lot of
Or about probiotic. Okay, right. Yeah.
We have inulin, we have oligosaccharides, we have fructooligosaccharides. Then we can use this barley. All these are prebiotics.
Okay. Then he's asking, can we use human probiotics to dogs? Can we give them?
I'm not sure of these dogs and bovines, what we can give to them. I am a veterinarian, but I'm very far from the field area right now, clinics area.
Then Dr. Swarna is asking, effect of processing on metabolomics compounds in milk.
Oh, great. This is very nice question. Right now, I've told you already, processing techniques, this involves pasteurization, sterilization of the milk, or any other method, HTST, LTLT. So pasteurization, there was one research paper which I have seen, and I have told you also that it is stable. It is having stable metabolome during initial storage up to 3 days, and then there is loss of vitamins and all. For regarding sterilization and all, there has been some loss of vitamins and essential amino acids during the later stages. So research is going on in this aspect also. We are also doing this effect of processing conditions on this. So let us see how it goes.
Okay. Okay, then there are other congratulatory messages to you, Dr. Heena.
Thanks.
And I would also like to ask that at what frequency can we take probiotics? Means a healthy person.
I think once in a week, what I do.
Okay,
Yeah.
okay. Right. So now I think we will close it, and I would like to formally thank you for your very interesting lecture. And it was not only from the veterinary or animal husbandry point of view, but since it was very much relevant to us, so everybody must have got benefited. And we really feel more knowledgeable at the end of this lecture.
Thank you.
And you have means the lecture content and the theme of the lecture, so it highlights the contribution of the veterinary and the animal husbandry professionals in uplifting the health and economy of the nation as well. And so I would just like to say thank you very much for sparing your valuable time, and the participants, I am confident they must have benefited from this lecture. Thank you once again.
Thank you. Thank you for inviting.
Thank you, everyone. Let's join back tomorrow at 11:30. 11.